Hello, everyone. Welcome to the Basic Science Oral Abstract session number 2. I will be chairing this session. I, Nicole Bernard, will be chairing this session with Dr. Jimmy Dikiakos. And I will be presenting the first 4 presenters, and Dr. Dikiakos will do the second. So the first presenter is Hannah Wilcox, who will give a presentation on neovaginal microbiome of transfeminine individuals. It's all yours, Hannah.
Oh, sorry. Okay, so hi everyone. Again, my name's Hannah Wilcox, and I'm going to be sharing my team's research on the neovaginal microbiome of transfeminine individuals. So transgender individuals, these are individuals whose gender identity does not align with the sex they were assigned at birth, and they make up about 0.2 to 0.5% of the adult population. However, until recently, they've largely been excluded from sexual health research. So we know, based on a number of large longitudinal studies, that the cisgender genital microbiome plays a really critical role in genital health, including susceptibility to sexually transmitted infections, including HIV. So today, this research is solely focused on transfeminine individuals, and these are individuals who are assigned male at birth but do not identify as male. And all the participants in our study are individuals who have undergone what is known as vaginoplasty, and this is a surgery to create what is known as a neovagina. So vaginoplasty is a form of gender-affirming surgery. And the most common form is penile inversion vaginoplasty, and it's actually the only surgical technique that's performed in Canada. So with vaginoplasty and penile inversion vaginoplasty in particular, you have dissection of the space between the rectum and the bladder, and then insertion of the penile tube tissue up into this space. So we end up with an organ that's very similar functionally to the cis female vagina. However, on a microstructure level, it's very different, as it is quite often lined with penile tissue, and these individuals don't have a cervix that's having cervical secretions, etc. So the aim of this research is to better characterize the neovaginal microenvironment, as it is most likely going to be a critical regulator of sexual health, including HIV acquisition. So as I mentioned, previous large longitudinal studies at the cisgender genital microbiome have found that an increase in the abundance of anaerobic bacteria is associated with both an increased risk in HIV acquisition and an increase in the local levels of HIV target cells, such as Cd4 positive T cells. So this is what we think is probably the likely mechanism of HIV seroconversion based on having a higher abundance of anaerobic bacteria in the genitalia. So here we have a bar plot that's generated from previously published microbial data from our group on both the cis female vagina and the cis male foreskin. So we can see right off the bat that oh, I should mention each bar is an individual sample, and all the various colors represent bacteria at the genus level. But right away, we can see that the vagina tends to be dominated by lactobacillus, and lactobacillus is very beneficial in the vagina. But we do have this more high-risk group in the vagina that has a more diverse set of anaerobic bacteria, as well as an increase, especially in Gardnerella, which you can see here in turquoise. In the cis male foreskin, we have a very diverse group of anaerobic bacteria, particularly a lot of Prevotella, Peptonophilus, and Porphyromonas, all these sort of green tones here. But we then have this sort of lower risk group in the cis male foreskin, and there tends to be an increase in the abundance of Corynebacterium in this group. The main point I want to drive across in this slide, though, is that what constitute high risk or low risk between the vagina and the cis male foreskin is very different. So this is important when we're moving forward and thinking about a structure that's created with penile tissue, but is sort of a vaginal structure functionally. So This research is part of the larger Transbiota study, and the Transbiota study aims to characterize both the transfeminine neovagina and the transmasculine vaginal microenvironment. So our participants are recruited through social media, through their healthcare professionals, and many of them also took part in this survey called Transpulse Canada, and they have been recontacted to take part in this study as well. So once they enroll online, they are mailed an at-home sampling kit, which has enough materials in it to take one sample per week for 3 weeks. We currently have 174 transgender individuals enrolled, and 57 of them are transfeminine. So of the 57 transfeminine individuals, I have 36 in the analytic population that I'll be showing you today. To enroll, our transfeminine participants must have undergone vaginoplasty at least 1 year prior to enrollment, and they also must have been at least 18 years of age. So we actually have a really diverse age group from 23 to 67 in this cohort, and also a really wide range between the time that they got vaginoplasty from 1.2 to 19.2 years ago. We also take in information via survey about any sort of hormone therapy these participants are on. You see that estrogen is a big one for these participants, and any treatments they might be taking or any symptoms they might be feeling when they take the swabs at home. So once we have the samples back in the lab, they get mailed back to us. They undergo bacterial analysis, and the bacteria is characterized via sequencing of the V3 to V4 region of the 16S rRNA gene. And like I said earlier, all the data I'm showing you today is just at the genus level. So for statistical analysis, we designated Lactobacillus dominans as a relative abundance of greater than 50%. We then defined Prevotella, Gardnerella, and Corynebacterium prevalence as a relative abundance of greater than 20%. And then proportions of the bacterial dominance or prevalence were compared across groups via chi-squared tests. So here is that same exact bar chart I showed you from earlier, but now we see the neovaginal microbial data here. So we can see right away that the neovaginal microbiome really doesn't look anything like the cis female vaginal microbiome. We don't see nearly that lactobacillus dominance that we see in the cis female vagina here. However, although the neovaginal microbiome tends to be a lot more diverse, it appears to be very different than the more high-risk group in the vagina. Specifically, we don't see nearly that abundance of Gardnerella in the neovagina that we see over here in the vagina. And overall, we do see that the neovagina looks somewhat more similar to the cis male foreskin, but there are some really distinct differences, such as that abundance of Corynebacterium that we get in this low-risk population in the cis male foreskin. We don't see a lot of that Corynebacterium here in the neovagina. We also see an increase in Ezekiella and Fusobacterium here in the neovagina, as well as an increase in Streptococcus, which is interesting. And I'd also like to note that we do see a really similar proportion of Prevotella between the cis male foreskin and the neovagina, as well as between the high-risk population in the vagina and the neovagina. And that's interesting to consider because Prevotella is one of these bacteria that's associated with HIV or increased HIV acquisition in both females and males. So here's just sort of a statistical summation of that graph. Once again, we can see that the lactobacillus dominance is very statistically different between the neovagina and the cis female vagina, with 72% of individuals having lactobacillus dominance in the cis female vagina, as opposed to only 3.9% of samples having lactobacillus dominance in the neovagina. Once again, we also don't see any Gardnerella prevalence in the neovagina as compared to the cis female vagina. And the same goes for Corynebacterium in comparison to the cis male foreskin with the neovagina. However, there isn't actually any statistical difference in the proportion of Prevotella prevalence between the neovagina and the cis male foreskin. So that's really interesting to note because, as I mentioned, Prevotella is definitely implicated in HIV seroconversion in, or associated with HIV seroconversion in both men and women. So, to conclude, the neovagina has a really unique bacterial composition that's distinct from both the cisgender genital microbiomes. We see that the neovagina has greater than 50% of the samples had a high prevalence of Prevotella. And as I mentioned, Prevotella is associated with HIV acquisition in both the vagina and the penis. Bacteria that were associated with low risk in the vagina of the penis were not found in the neovagina to any great extent, and this is lactobacillus in the vagina and Corynebacterium in the penis. So this descriptive study is a really huge step forward in elucidating the microenvironment of the neovagina, but a ton of research still needs to be done. Although we can see these bacteria present, such as Prevotella, we don't really know if it's biologically relevant or if it's associated with negative health outcomes until we study this microbial data alongside immune data or symptom data. And then we can get a better idea of what bacteria are actually associated with negative health outcomes, such as HIV acquisition, and which bacteria are associated with a low inflammatory and healthy microbiome in the neovagina. So with that, I'd like to thank my supervisor, Dr. Jessica Prodger, as well as all the members of my lab for all their help and support. And especially I'd like to thank all the members of the Transbiota group, Dr. Greta Bauer, my professor, Dr. Jessica Prodger, Dr. Jacques Ravel, Vern Minari, and especially Jason Hillarn for all their help and support. And I'd really like to thank all our participants for lending their time and their vaginal samples to this study. And I'd be happy to take any questions.
Thank you very much, Hannah. That was a great talk. I may have missed it, but is the foreskin included in the surgery?
Pardon?
Is the foreskin included in the surgical procedure?
Oh, in the creation of the neovagina?
Yes.
Well, it just depends, I imagine, if they've been previously circumcised or not, but that's definitely a question that's come up previously, like, would there be a difference if they were circumcised prior to vaginoplasty as opposed to if they weren't? But that just depends on the participant, really.
So we have some questions from the audience. A few of your neovaginal participants do show a lactodominance. What do you think is causing or driving that? So
we don't really know, and this is quite early, but we have found that it seems that the participants, I should back up, in the vagina, there tends to be not just lactobacillus dominance, but dominance by 4 lactobacillus species in particular. And we actually just recently found that it seems that those samples that have lactobacillus dominance are actually the different lactobacillus species. So that's really interesting. And it could be that maybe they were taking some sort of supplement around the time of sampling, but these are the things we need to look into in more in greater detail and really fine comb through all these things. But there could be any number of reasons why we're seeing that lactobacillus dominance in those groups in particular.
We have a question here from Dr. Poon. Great talk, he says. Will you do a clustering analysis on your data, or have you already?
Yeah, we are planning on doing clustering analysis with our data, as well as the immune data we are gathering from this project. But we are planning on doing clustering analysis to sort of define potential community state types that exist in the neovagina.
And we have a question from Christina Hayes. What database did you use to assign taxonomy to your V3, V4 sequencing?
So that's actually done by our collaborator group in Maryland, but they use Pecan and Speciate, and then they reduced the species data to a genus level to align with our vaginal and cis male foreskin data. Okay, I don't see any more questions. Let me take another look. I think that is it. Are there any questions from the panelists? If not, thank you very much, Hannah. That was great. We'll move on to the next talk that will be given by Mitchell Mumby. And the title of the talk is Characterizing the Intracellular Trafficking Pathways Hijacked by HIV Neph to Downregulate SERIC5.
Okay, thank you very much. As always, it is a pleasure to speak in this forum, and I'm always very much grateful. Okay, To introduce, HIV virions will readily egress from the surface of an infected cell, as depicted here. And following virion maturation, these particles will then infect a target cell by binding to the CD4 chemokine co-receptor. This creates a membrane fusion event, which deposits the viral capsid in the host cell cytosol and thereby permits replication to commence. However, in the presence of the serine-5 restriction factor, we see as these virions egress, serine-5 is readily incorporated into the outer lipid membrane, such that when these particles attempt to infect a target cell, membrane fusion is blocked, which thereby prevents deposition of the viral capsid and thereby lowers virion infectivity. Now, to counter such a selective pressure, the HIV accessory protein Neph triggers serine-5 endocytosis from the cell surface, and it does so by interacting with adaptin protein 2 or AP2. So endocytosed serine-5 is first directed to the early endosome, and it is believed that it is ultimately directed to the lysosome where it is degraded. The question is whether Neph interacts with other APs to facilitate this trafficking, and whether it goes into the late endosome multivesicular body pathway, or perhaps into this trans-Golgi network compartment, which is a common feature of other Neph functions. What's interesting is that Neph downregulates the HIV entry receptor CD4 from the cell surface in a manner that's similar to serine-5 downregulation. In both cases, it is believed that the N-terminus of Neph is what is selectively interacting with serine-5 and CD4, where the C-terminus is interacting with AP2. And Neph does this by supplying its own Neph-dileucine motif, which interacts with the sigma subunit of AP2, and Neph forms additional peripheral interactions with the alpha subunit. Now, when we look at CD4 downregulation, in a manner similar to serine-5 downregulation, there is this Neph-AP2 interaction that directs CD4 from the cell surface and into the early endosome. From there, Neph interacts with AP1 gamma 2, which is an AP1 complex bearing the gamma 2 variant subunit, and this directs CD4 into the multivesicular body pathway, where it is eventually degraded within the lysosome. What's interesting is that interactions with AP1 gamma 1, the canonical AP1 complex, is not required for this particular function. And so to explore this trafficking pathway between serine-5 and CD4 a little bit further, we acquired two primary Neph isolates, 2410 and 2391, and assessed their ability to downregulate cell surface serine-5 and CD4. And so in this assay, we had CD4 positive HeLa cells, which are co-transfected with our plasmids encoding for serine, along with plasmids encoding for our primary Neph EGFP and our EGFP only negative control. Following 24 hours, the cells were then surface stained for serine-5 and CD4, and then flow cytometry was used to quantify the cell surface levels of either protein. And so when we look at CD4 downregulation first, we see that isolate 2410 and 2391 robustly downregulate CD4 from the cell surface. And interestingly, when we look at their dileucine mutants, where these two leucines are mutated to alanines, which severs the Neph-AP interaction, we see that this functional ability is subsequently abrogated, and this is known in the literature. When we look at serine-5 downregulation, on the other hand, we see that isolate 2410, but not 2391, downregulates serine-5 efficiently, and accordingly, the respective dileucine mutants were also defective in this function, which would indicate that this is also an AP-dependent function like CD4. Now, it's interesting to note that we saw these functional differences despite the fact that both of these constructs were expressed to similar levels, and this was determined via flow cytometry as well as Western blot, as shown here. And so this led us to our hypothesis that 2391 Neph must encode a polymorphism that is able to functionally uncouple its ability to downregulate cell surface CD4 and serine-5. Now, to answer this question, we conducted a series of chimera Neph experiments where we swapped a region of 2410 with 2391 Neph and assessed cell surface serine-5 and CD4 downregulation ability until we eventually came across the polymorphism of interest. Now, I'm not going to show this data. It's very repetitive. And for the sake of brevity, I'll just say that this polymorphism was ultimately mapped to the ND164 region in 2391 Neph. And as can be seen here, this is located within the Neph-dileucine motif, which was a bit of a surprise for us. Indeed, when we look at CD4 downregulation in this case, we see that 2410 that bears this ND164 polymorphism suffers a modest impairment in CD4 downregulation ability relative to 2410. But while this is not significant, there's a partial impairment, but it's important to note upon comparison to EGFP only that this is still able to downregulate CD4 to some extent. When we look at serine-5 downregulation, on the other hand, we see that the ND164 polymorphism this time results in a much more severe reduction in serine-5 downregulation ability, and this is evidence upon the equal level of downregulation as compared to the EGFP-only negative control. And so to schematize this, we see that in the presence of 2410 Neph, we're getting downregulation of both CD4 and serine-5, but in the presence of ND164, we're getting just CD4 downregulation with minimal, if not any, serine-5 downregulation. Now, as I mentioned previously, downregulating serine-5 is intimately related to the infectivity of the viruses. And so we would like to determine whether the ability of these Neph proteins to downregulate serine-5 corresponds to an associated effect on infectivity, or in this case, infectious virus yield. And so in this experiment, we triply transfected HEC293 T cells with our plasmids encoding our Neph samples, along with GAG plasmid, as well as serine-5 or with the PBJ5 plasmid. And so this would represent the presence of serine-5, and this would represent the absence of serine-5. Following 48 hours, we collected our viruses. So we can see our viruses that do not have serine-5 incorporated, and then our viruses with serine-5 incorporated. And then we infected TZMBL cells for 72 hours. And so for those that do not know, TZMBL cells are basically just HeLa cells that have the luciferase gene under the transcriptional regulation of the HIV LTR. And so in this case, relative luciferase units would be proportional to the infectious virus yield of our particular sample. And so from that, we then calculate relative infectious virus yield by taking our mean relative luciferase units in the presence of serine-5 compared to the mean RLUs in the absence of serine-5. So in this situation, because 2410 we know downregulates serine-5 the most efficient from the cell surface, we would expect it to have the highest RLUs in the presence of serine-5 and therefore have the highest relative infectious virus yield. And so indeed, when we look at relative infectious virus yield, relative to the delta Neph sample, we see that not surprisingly, 2410 Neph does indeed produce the highest relative infectious virus yield. When we look at the ND164 polymorphism, we see that there is a significant reduction in relative infectious virus yield compared to 2410, but importantly and consistently with our previous experiments, this was about equal to our delta Neph negative control. And furthermore, we noticed that once again, our dileucine mutants that I mentioned previously, as well as 2391 Neph, also feature this sort of more significant reduction in relative infectious virus yield accordingly. And what this ultimately tells us is that the ability to downregulate serine-5 is something that we can measure by looking at infectious virus yield as a functional output. Now, because this ND164 polymorphism occurs within the Neph dileucine motif, and we know that this is important for Neph AP interactions, the next logical step would be to determine which APs Neph interacts with in order to downregulate serine-5 from the cell surface and to the lysosome. And so what I propose for these experiments that we're currently still working on, we're going to treat our CD4 positive HeLa cells with siRNAs that are targeting the large subunits of a variety of APs that might be implicated. So we're looking at, first of all, the alpha subunit of AP2, which we think is definitely involved because of its relationship with the cell surface. We're also going to be targeting the gamma-1 and gamma-2 subunits of AP1 to delineate between the variants of the AP1 complex. And then we're also going to look at the large delta AP3 subunit accordingly. And after 48 hours, these our CD4 positive HeLa cells will then be co-transfected with plasmids encoding for serine-5, along with plasmids encoding for 2410 and its respective dileucine mutant. And from there, we will assess cell surface levels in a manner I described previously and measure via flow cytometry. And so because we think AP2 is involved, we expect that silencing the alpha subunit would result in more serine-5 on the cell surface in the presence of 2410 Neph, which would indicate that there's now some impairment in its downregulatory ability. So this is kind of the output that we're going to look for. And from this, we can use our ND164 polymorphism to figure out exactly how it's affecting its relationship or interaction with any implicated APs. Okay, so to summarize, this ND164 polymorphism is a clinically relevant polymorphism. This is actually from a figure from my paper where we looked at all the curated Neph sequences at the Los Alamos database. And we found that aspartic acid at this position is present about 0.39% of the sequences, which may not seem like a lot, but when there's thousands of sequences that are deposited, this could be somewhat relevant in an absolute sense. Furthermore, this does add to a growing body of evidence that serine-5 and CD4 downregulation are genetically separable. And then furthermore, we will use our ND164 polymorphism mutant to further understand how Neph can selectively target CD4, but not serine-5. And finally, this could allow for us to design potential compounds that can target these complexes in a more specific manner. And with that, I would like to primarily thank my wonderful supervisor, Dr. Jimmy Dikiakos, as well as my advisory committee members, all of my past and present lab members, as well as my funding sources. And with that, I would be happy to take any questions. Thank you.
Oh, thank you. That was an amazing talk. Let me see if there are any questions that have come in. Not yet. This system is not very familiar to me, so I may be asking a question that is not very relevant to your presentation, but I know that Neph also down modulates HLA, if I'm not mistaken.
Yes.
So is that something that's part of the plan to look at these variants in terms of HLA down modulation, in addition to Serink and CD4?
You know, we haven't actually, planned this concretely, but I think that's actually a really interesting idea because, with MHC1 downregulation, you're using different APs as opposed to CD4 because there's a couple different mechanisms by which that one occurs. So if we're seeing that this ND164 polymorphism is actually affecting the affinity to AP complexes, yeah, it would be very curious to see whether that would have an impact on MHC1. I think that would be a logical thing to do after we've kind of figured out the APs that are involved, because it's becoming clear that it's much more complex than many originally thought.
I have a question here from Zabrina Brume saying, fantastic talk. Do you have plans to look at Serink 3?
That's a good question. We have not, and I know that Serink 3 and Serink 5 downregulation by Neph is also genetically separable. I believe it was Zabrina's group that showed that. And there's a couple really nice papers that they published that shows a lot of the polymorphisms that are associated with either or. So I think that once this whole picture has been solved for us, I think that's another thing that we should look at too, for sure.
Any members of the panel have questions for Mitchell? Let's see. Yes, there's a question from Keith F. I don't know who that is, but does Neph function as a dimer, and how does the polymorphism affect dimer function?
So the answer to that is yes, Neph does function as a dimer. It really depends on the context whether it's relevant for a particular function. So I know for a fact that Neph dimers are involved, or at least have been suggested to be involved with Serink 5 downregulation. At this point, because there's no crystal structure of Serink 5 with Neph, it's pretty hard to ultimately kind of speculate on that question, because we don't know if Serink 5 even exists as a monomer or potentially as a hexamer, which has been suggested in one of the previous publications. And so if that's the case, then we have to really think about how these APs are kind of organizing themselves. What comes to mind is when when SIV Neph downregulates tetherin, there's a much more complex organization involving 3 AP complexes. So that's a question I really don't know the answer to yet. I think once we facilitate some of these in vitro binding assays, see whether this polymorphism is affecting in a one-to-one stoichiometric ratio. If that's the case, then we can kind of make more suppositions moving forward about how this complex comes together.
Okay. I don't see any other questions. So if there aren't any, we'll move on to the next talk, but great job.
Thank you very much.
So the next presenter is Abayomi Olabode. I'm sorry if I'm mispronouncing your name. And he will be talking about phylogenetic reconstruction and in cellular function characterization of the ancestral Neph protein of primate lentiviruses.
Thank you. I'm going to be talking about the phylogenetic reconstruction and in-cellular functional characterization of ancestral Neph protein of primate lentivirus. Now, lentivirus Neph are newly emerged genes found only in primate lentiviruses, and they encode a small protein that is disordered and interact with a wide range of cellular proteins that perform multiple functions. And they play a vital role in viral pathogenesis, such as antagonizing serine C, like Michelle just talked about, to enhance viral replication, and deregulating CD4. So the objective of our study was to reconstruct the ancestral Neph sequence of primate lentiviruses and study those sequences in a structural and functional context. So one of the things we've done was to get 34 primate lentivirus sequences consisting of HIV and SIV, and aligned them, reconstructed them, built a maximum likelihood tree, and then we rescaled the trees back in time under a relaxed clock using BEAST, and then we did an ancestral reconstruction to reconstruct the sequence states of the internal nodes of the trees. And then we did a structural analysis to study the history of intrinsic protein disorder in these ancestral viral Neph lineages. And then we sent the sequences on the trajectory to the HIV-1 group M part to the CAHR lab, a work which Mitch did. So he's talked about this procedure. I won't spend more time on it. But basically, they synthesized the nucleic acid and cloned them into expression vectors, and then co-transfected serine C and Neph, or EGPF, into CD4 HeLa cells. And after 24 hours, they stained the cells to capture CD4 or serine C, and then measured using flow cytometry. So here are some results talking about the distribution of intrinsic disorder in modern day primate lentiviruses. This is an example of what the Neph protein looks like. And we know that the N-terminal and anchor region and sometimes the C-terminal domain have been shown to be flexible. And a protein is called disordered when that region of the protein is flexible and unstructured, whereas the core domain is always conserved and well-folded and structured. So on the right here, we have the phylogeny of the present-day Neph sequences, and the heat map here shows the distribution of intrinsic disorder, and red here indicates disorder, whereas the blue indicates that those regions are structured. So here we can see the distribution varies across sequences and is consistent with the phylogeny. The white spaces here just indicates that those are regions where there are indels between sequences. Then next, we wanted to look at some of the ancestors of ancestral sequences of Neph to see if they were look at the history of intrinsic disorder. And we looked at the sequence at the root, the ancestor of HIV2, the CPZ ancestor, and then the ancestor of HIV-1 group M. And what we're showing here is that the ancestors of HIV group M and CPZ are getting disordered over time compared to the sequence at the root or the ancestral sequence, whereas HIV2 is getting less disordered over time. And we went ahead to look at the impact on length, sequence length as well, and we see the opposite, where HIV and the CPZ ancestors, HIV1 group M and CPZ ancestors were getting shorter over time, whereas HIV2 was getting longer. Then we looked at some already characterized functional motifs on the ELM database to see if some of those motifs are present in our ancestral lineages and if they've been conserved over time. So here red or 100 means that for 1,000 random beast trees and for that lineage we're looking at, the motif is present 100% of the time or 0% of the time, and then we have intermediates. So, and on the right here, we have a dendrogram that just clusters motifs based on unrelatedness. So we can see that there's a region where some of those motifs are present and conserved all through time. For example, we have the end myristylation site and the proline-rich motif, which are commonly studied. And we also see that there are some motifs that emerged that are not present in the root, but emerged in CPZ and HIV lineages and are not in the HIV2 SMM lineage. And also there are some of those motifs that were in the root or ancestor and lost in CPZ and HIV1 group M. Next, we sent out sequences on the ancestral part to HIV group M, HIV1 group M for functional characterization, where we were looking at CD4 and serine C. That we did a CD4 and serine C down regulation assay using CD4 LR cells expressing the ancestral Neph. We reconstructed and some control sequences such as SIV MAC239 and some HIV primary Neph isolates 2410 and 2391, which Mitch just talked about, and serine C. And then we stained those cells using, we stained those cells, the CD4 and serine C, and then analyzed them using flow cytometry. So here we're looking at serine C down regulation, and we're looking at down regulation in by comparing to the negative control, which is GPF, where there's no Neph. And here in the already characterized 2-4-10 GPF, we can see that there's a threefold down regulation compared to GPF. And then in our ancestral Neph, which we reconstructed, we can see that this sequence here is able to downregulate serine C5. And we also looked at its impact on CD4 down regulation. And here we're also showing that though our ancestral Neph downregulates serine C compared to GFP only, it does that poorly, and it's that our capacity to downregulate compared to the 2-4-10 Neph is reduced threefold. And then this image here is just to show the expression levels relative to GPF, and we can see that our ancestral Neph is also well expressed. Next, we wanted to then look at other intermediate sequences on the ancestral trajectory to HIV1 group M, and we see that for serine C downregulation, our ancestral Neph is able to downregulate it, but that function is lost in the intermediate ancestral Neph sequences, and then it emerged just before the ancestor or ancestral Neph of HIV1 group M. Similarly, for CD4, we see that the ancestor does poorly, but and then also the next two intermediate ancestral Nephs didn't do much in downregulating CD4, but then it begins to peak until the ancestor of HIV1 group M. And here we're just showing the expression levels again from all our constructed ancestral Nephs, and we see that they are expressed in levels that are comparable. But in order to determine if we're just measuring GFPs or Neph sequences that are fused with GFP in our flow cytometry, we also did a Western blot to confirm that these constructs were there, and we find them in the right sizes here in this heat map, and we find some consistency in the expression levels as well. For example, in this control SIV Mark 239, you find that it's lowly expressed here, and in the Western blot is also lowly expressed. So this kind of gave us some confidence. So in conclusion, we were able to successfully reconstruct the primate ancestral Neph, and we find that they were viable and functional. And here we're showing a workflow for resurrecting and studying ancestral proteins that may be useful. And then we showed that Cd4 down regulation slowly evolved over time, whereas serine C was in the ancestor, it disappeared and then emerged just on the ancestral part to HIV1 group M. And then we can describe the history of Neph in two major lineages, one that with genomes becoming more disordered over time, and another with genomes becoming less disordered over time. I would like to thank my boss, Dr. Poon, for his support, and the CAHR lab, Dr. Jimmy and Mitchell, that did most of the experimental work, and the entire Poon lab. Thank you.
Thank you. That was very nice work, nice presentation. So from what I understand, when you go back in time to the ancestral Neph and the root Neph, that you see an uncoupling of the downregulation of serine 5 and Cd4 downregulation. What would be the implication of that? Like, how would that affect perhaps the fitness of the virus, or how well it functions or replicates?
Yeah, so one of the assumptions we have in is, for example, the serine C downregulation was lost over time and then emerged just before the ancestor of HIV1 group M, which became the most successful of all the HIVs that infects humans. And it's over 90% of cases of HIV are actually from HIV1 group M. And so one thought it could be that that ability to downregulate serine C was actually very important for the success of the virus. And so that might have contributed to its fitness or its ability to thrive, actually.
Can you speculate on how it would affect its fitness?
I can't say, but sorry. But then I think that it's possible that it improved the fitness, because we're looking at down regulation of serine C, and serine C, in the absence of serine C, the virus is able to replicate more. But yeah, I can't speculate 100% on how much it improved the fitness. I think Mitch and Dr. Jimmy might know better.
Do you want to add anything to that, Mitch or Dr. Dikiakos?
Mitch, you can go ahead if you like.
Sure. So we think that it's pretty well known that tethering was always kind of the species barrier for transmission from chimpanzees to humans. So we're not sure whether serine 5 evolution became, something that was required for cross-species transmission. We've kind of always looked at it as serine 5 downregulation evolved to disseminate widely within a host population. So it's maybe not necessarily required for transmission purposes. And if transmission is ultimately high, there would be no need for serine downregulation to occur, at least in the same extent. But if transmission is quite inefficient, at least in the early stages of human transmission, we speculate that serine 5 would have evolved rather quickly to kind of compensate for those mechanisms.
Interesting. Okay, anything to say about the CD4 not being downregulated by the ancestral forms?
I have no idea.
Okay.
The thing is, the CD4 is that it's pretty similar amongst all the simians, at least from what I can tell from available data. So I'm not really sure whether that would be selected for per se, because the thing is CD4 downregulation by Neph is it does so by preventing ADCC and superinfection ultimately. So I would think that it would have to have something to do with that, where if those two factors weren't as important, perhaps they wouldn't necessarily have evolved in such a manner as if they were absolutely required.
Okay, well, there's some questions that have come in from the audience. So regarding fitness, this is from Dr. Poon. Regarding fitness, I think it's difficult to say because the virus is moving between host environments. So that's somewhat similar to what you said. And from Sabrina, thank you for the great talk. Do you think that the functional results would be similar if you used a non-human cell line? The very far back ancestral Neph would not have replicated in humans.
Yeah, so I think it's possible that we would get different results until we tried that. I think it's possible, but I think the HeLa cells were used because of the stable cellular network and how it was easier for us to combine studying CD4 and serum C. Yeah.
Great, thank you very much. I guess we'll move on to the next presentation, which will be by Corby Fink, who will be talking about the adjuvant role of Neph inhibitors towards a cure for HIV/AIDS.
All right, hi everyone. So I will be speaking about obviously the adjuvant role of Neph inhibitors towards a cure for HIV/AIDS. So despite currently available and effective therapeutics, HIV-1 persists due to its ability to evade the immune recognition. One second. So sorry, its ability to evade immune recognition. So for example, HIV is able to establish latent or minimally replicated tissue reservoirs. And because of this, purging of this reservoir as a candidate reactivation strategy has been suggested. However, with reactivation of HIV comes expression of the accessory proteins VIF, VPU, VPR, and Neph, the latter of which will be the focus of my talk today. So with Neph expression comes Neph-mediated immune evasion, which is currently one of the contributing factors that precludes development of a functional cure. So Neph has a multifactorial way in contributing to HIV pathogenesis. It can alter cell trafficking pathways to subvert apoptosis, enhance viral replication, as well as suppress CD8 positive cytotoxic T lymphocyte or CTL mediated immune responses. So in this schematic here, Neph shown in green is using host proteins is transported to the trans-Golgi network or TGN, where it interacts with Sarc family kinases or SFKs shown in blue. This initiates a cascade of events that includes multi-kinase complex formation, but ultimately culminates in MHC class 1 downregulation from the cell surface and sequestration at the TGM. So with this in mind, we thought of conducting a screen to identify small molecules that are able to inhibit this Neph-SFK interaction and thus rescue Neph-mediated MHC downregulation. So our group has previously identified an inhibitor termed compound 2C that binds Neph in a putative binding pocket with Neph shown here in green and SFK in blue. We used a different in silico drug discovery screen to identify compound H31, which is the compound that I will be speaking about today. So compound H31 is a histidine tyrosine dipeptide that's predicted to inhibit this Neph-SFK interaction through its high affinity binding to the SH3 binding domain. So because of this improved binding affinity, it's possible that H31 may possess an improved in vitro toxicity profile compared to a progenitor compound 2C. So with our compound H31 included in the same schematic, we predict that H31 will inhibit or disrupt this Neph-SFK interaction, which will then minimize or limit multi-kinase complex formation, downstream signaling, with our end goal then being a rescue of Neph-mediated MHC down one, class 1 regulation. So the overall goal of our work is to inhibit the Neph-SFK complex formation with H3.1 to act as a molecular adjuvant within HIV vaccine strategies. And to achieve this goal, we have 3 main objectives. Our first is to determine the capacity of H3.1 to rescue Neph-mediated class 1 downregulation. Our second is then to determine the optimal H3.1 administration route, dosing, and in vivo biodistribution in a mouse model. And lastly, we hope to assess the role of H3.1 as an adjuvant in a dendritic cell or DC-based vaccine reactivation strategy in a mouse model of HIV/AIDS-like phenotype. So to address our first question, to address our first objective, we asked the question, do our inhibitors rescue MHC class 1 downregulation? So to assess this, sub-T1 cells were infected with lab-adapted NL4-3 expressing EGFP, after which we added either progenitor compound 2C or our compound H3.1 to culture. Following the completion of culture, EGFP expression served as a readout of our infection. And within the EGFP positive population, it was further subdivided into EGFP low, medium, or high expression, just shown here in this box dot plot. So we use these same populations to then quantify surface MHC class 1 expression, which for each group is being represented as a fold change from uninfected cells of the same condition. So what we saw from this experiment is that regardless of which specific subpopulation of EGFP cells we're in, that we were seeing approximately a 10% rescue in surface MHC as a result of H3.1 addition to culture. And additionally, in this specific experiment, we did not see a rescue associated with compound 2C, and that they're obviously one that was noted with our H3.1. So after we were able to observe that this MHC expression is rescued following H3.1, we then asked the question of if this MHC rescue is Neph dependent. So in a similarly designed experiment, sub-T1 cells were either infected with NL4-3 EGFP, which is in the top row here, or NL4-3 EGFP lacking Neph, or NL4-3 delta Neph. So what we can see in this, and then we assess the MHC surface expression using MFI and flow cytometry. So in our top row here in our top histogram, for cells infected with NL4-3 containing Neph, what we saw is that increasing concentrations of H3.1 resulted in increased rescue of MHC class 1 on the cell surface, albeit not to levels seen with uninfected cells, which would be the black histogram here. In contrast, for cells infected with NL4-3 lacking Neph, regardless of the concentration of H3.1 that we added, we were unable to see a rescue of Neph-mediated downregulation. So collectively, what this data tells us is that the H3.1 mediated rescue that we see of MHC1 surface expression is both dose-dependent and Neph-dependent. So we then moved on into our in vivo CD4C HIV Neph transgenic mouse model. So this mouse model contains a murine CD4 enhancer, a human CD4C promoter, the NL4-3 coding, and an SV40 poly-A tail. So it's important to note in this model that the upstream sequence dictates tissue Neph specificity, which in this specific model accurately models Neph expression as would be observed in HIV-infected humans. So these mice have a measurable decrease in CD4 T cells by approximately 3 weeks of age. And in most animals at around just over 1 to 2 months, we can observe an AIDS-like phenotype. So because this model that we chose accurately recapitulates what's seen in humans, it allows us to assess 2 important points within our study. So our first of which is to assess the biodistribution and in vivo stability of H3.1. And in this way, we will be able to assess and optimize certain parameters like administration route, H3.1 dosing, potential toxicity that may arise, as well as measuring H3.1 concentration within the plasma. And with the same animal model, we can also assess MHC class 1 and antigen presentation through ex vivo culture and addition of H3.1. So with respect to our H3.1 biodistribution and stability, our animals were administered this compound in a range of concentrations from 8 to 32 milligrams per kilogram, either through oral gavage or intraperitoneal injection. Then at predetermined endpoints, the animals were sacrificed, organs were collected for future toxicity assessment, as well as collection of blood such that we can measure plasma H3.1 concentrations using mass spectrometry. So what we see in our summary graph here is that for male mice in green or female mice in purple that received H3.1 through intraperitoneal injection, we were able to detect plasma H3.1 at the earliest time point of collection. However, from 30 minutes onward, the concentration of H3.1 in plasma was below our detection threshold. Additionally, for male or female mice, it didn't matter, that received oral lavage administration of H3.1, we were unable to detect H3.1 within the plasma using mass spectrometry. So the take-home message from here is that H3.1 is rapidly cleared in vivo, and that this rapid clearance is most likely due to proteolysis of H3.1, given its dipeptide structure. That being said, when we looked at H3.1 in an ex vivo setting, we did see some interesting trends that warrant future investigation. So these trends were seen more with MHC class 1 and antigen presentation. So in this experiment, we took either wild type mice or a Cd4C transgenic mice, ex vivo cultured splenocytes with increasing concentrations of H3.1. Additionally, because we chose to breed these mice on a C57 Black 6 background, we are able to then add a commercially available and immunogenic class 1 restricted OT1 peptide to assess the presentation of this peptide in the context of MHC class 1. So just to quickly show the stark differences we see in our animal model, I'm going to draw your attention to the top right here. So in this top right FACS dot plot, this is what we see in our wild type Black 6 mice, where we can see a very clear defined CD3, CD4 positive T cell population, which is almost completely absent in our transgenic mice. So after selecting for these CD4 positive cells, what we then did is use an antibody and flow cytometry to assess the presentation of this OT1 peptide in complex with MHC class 1. So for wild type mice, it didn't matter based on incubation time from 120 minutes up till 360 or concentration of H3.1 in culture, we saw consistent surface presentation and expression of this complex. In contrast, when we look at our CD4C transgenic mice, an increase in incubation time trends to have more expression of OT1 complex to MHC class 1. And additionally, these studies are showing that with increasing concentrations of H3.1, we're trending to even further increase presentation of this peptide complex. And we believe that these results that we are seeing warrant, again, further investigation. So what I've shown today is that this compound H3.1 is able to inhibit Neph-mediated MHC class 1 down regulation. It's rapidly cleared following in vivo administration, and it also enhances presentation of immunogenic class 1 restricted peptide in ex vivo culture. So our current and future studies are dealing with the synthesis of H3.1 analogs that hopefully have better in vivo and in vitro characteristics. And all of this work is spearheaded by an MSc candidate in our laboratory, Anthony Lurie, and I encourage everyone to check out his poster in the Basic Sciences tract. Following synthesis of these analogs, we will then work on rigorously characterizing them, which will include but are not limited to assessing the ability to bind Neph, its ability to disrupt the Neph-SFK interaction, its in vivo stability, and then ultimately determining its role as an adjuvant to improve MHC class 1 antigen presentation. So with that, I would like to thank lab members, collaborators, and funding sources. Thank you.
Thank you. That was amazing. Okay, let me see if there are any questions that have come up. No, nothing yet for this. So I guess you're not too discouraged about the fact that the H3.1 doesn't last very long in the animals. You're just going to use that to build upon trying to improve that molecule. Do you have any idea? I mean, you mentioned that you had done some kinds of studies on the animals to look at, whether or not there's like a downside or pathogenic issues that may occur when you administer this molecule that would preclude going on to transferring this to a human system?
So I was trying to be careful to say that potential toxicity issues, we have yet to see them at high concentrations in these animals, where the process is still ongoing. So the toxicity wasn't the issue. The organs were collected in case we ever had to go back and look for them, but none were noted in the time points where we administered up to, I believe, 8 to 12 hours. So there was nothing, and that was when experimental endpoint was reached. So we didn't observe any toxicity yet.
Okay. Something else that I wanted to ask you. So in one of your last slides where you were looking at T cell responses, you were looking at MHC expression levels when you treat the cells with this molecule in vitro. Was that there was one slide where you were showing a flow plot, but I can't remember what was on the x-axis or the y-axis, whether or not you actually did have T cell responses when you exposed your cells to your compound.
Right, sorry, so that it was CD3 on the X and CD4 on the Y. So we were just looking at standard CD4 positive T cells. Okay. So it wasn't a T cell response. We were actually just characterizing using flow cytometry. We selected for our CD4 T cells. And then on those CD4 T cells, we looked at the expression of peptide complex to class 1.
Okay, I understand. Okay, let me see if there are any questions. Have you thought about administering your H3.1 in nanoparticles, perhaps in vivo?
Yeah, so nanoparticles have come up different delivery methods as well. We've even looked in very preliminary discussions at using some sort of pump where it's administered continuously as one way of circumventing or extending the rapid clearance that we're seeing.
So if there are no other questions, I mean, that was a very well-constructed study, so very interesting. Good luck with it, continuing with it.
Thank you.
So I'm going to pass the baton to my co-chair to continue with this session.
Thank you very much, Dr. Bernard. It's my pleasure to continue with the final 4 speakers of this fantastic session so far. I'm really happy to introduce our next speaker, Natalie Kinlock from Simon Fraser University, who's going to tell us about genetic integrity during long-term ART. Natalie, whenever you're ready.
Cool. Thanks very much, Jimmy, for the introduction. So like Jimmy mentioned, the title of my talk today is In-Depth Characterization of Genetic Integrity and Longevity of HIV Proviruses that Persist During Long-Term ART. So during active untreated HIV infection, HIV is continuously evolving, and members of that viral quasi-species are being seeded into the persistent HIV reservoir immediately and continuously over the course of untreated infection. However, not every provirus that's established into the persistent reservoir during this time is created equal. Only a small minority are genetically intact and are capable of reactivating from the reservoir to go on to infect new cells. And the remaining majority harbor a variety of defects that render them incapable of replicating, although in many cases, such proviruses are capable of producing RNA and/or proteins that can be recognized by the immune system and allow these persistently infected cells to be eliminated. So we know that these proviruses are entering the reservoir, but we know less about what happens to them once they're established there and how much that reservoir reflects the pre-ART evolutionary history of HIV in an individual following long-term ART. Although there's an increasing body of evidence that after long-term ART, the majority of the reservoir is comprised of viral variants that are seeded there closer to the time of ART initiation. However, we also know that the composition of the persistent reservoir is not static, and these changes are the result of dynamic cellular processes such as homeostatic proliferation, clonal expansion, and/or preferential elimination of specific persistently infected cells, such that the composition of the reservoir changes over time. Moreover, these processes may impact different subsets of the reservoir differently, as intact and defective proviruses have been shown to decay out of the reservoir at different rates. So the objective of our study is to increase understanding of the composition and dynamics of the persistent HIV-1 reservoir through in-depth sampling of individual proviral templates within host. So in order to do this, we first started by characterizing the proviral composition of 6 individuals living with HIV who are on long-term suppressive antiretroviral therapy, from whom we've collected more than 2,000 near full-length proviral sequences in total. What we observe, consistent with previous reports, is that the proviral reservoir is overwhelmingly defective, where on average 98% of near full-length proviruses we collected were defective, meaning that only 2% were genetically intact. Most individuals, proviruses with large deletions were the most common type of defect, making up between 39 and 90% of all proviruses recovered. Although participant BC003 is an exception to this, where this participant's reservoir carried a uniquely large burden of hypermutated proviruses. Despite these trends, the distribution of proviruses with specific defects was unique in each participant. Across participants, the GAG and Neph genes were the most commonly intact, where 31% of near full-length proviral sequences harbored in either intact GAG or an intact Neph gene. POL was the least commonly intact subgenomic region across study participant sequences, where just 18% of near full-length proviral sequences carried an intact POL gene. Clonal expansion in the proviral reservoir also varied from individual to individual, ranging from 17% in participant BC003 to 55% in participant BC027, where a median of 38% of near full-length proviral sequences per participant were part of a clonal set. The frequency at which these clonal sequences were recovered ranged from twice to 39 times, where in each individual, at least one heavily expanded clone was recovered, and where this most heavily expanded clone in each individual made up an average of 23% of clonal sequences and 8.5% of all sequences recovered in the individual. So for example, in participant BC027, the most frequently recovered clone was a sequence with a large deletion that was recovered 39 times. And this sequence makes up 36% of clonal sequences and 20% of all of the sequences that we recovered from this individual. Interestingly, also across participants, we noted that the burden of clonal expansion among both intact proviruses and those with defects in their packaging signal that are termed proviruses with 5' defects was significantly greater than other proviruses, where the odds of being part of a clonal set were 2.4 times greater for an intact provirus than other proviruses, and 2.9 times greater for proviruses with 5' defects. The burden of clonal expansion was significantly less in proviruses that were defective due to hypermutation, where the odds of being part of a clonal set was 0.47 times less than other proviruses. So what we next sought to understand is when these proviruses that have persisted in the reservoir of these individuals were actually seated there, and how that age distribution varies between individuals and between proviruses that have different genetic features that may impact their longevity. So we're able to do this in each participant for a subset of our proviral sequences that harbor an intact Neph gene, where we seek to interpret the diversity in these proviral sequences in the context of the pre-ART diversity observed in the individual. How we do this is first by collecting longitudinal pre-ART plasma HIV RNA and F single template sequences in order to reconstruct the evolutionary history of HIV in the individual. So in this example, plasma HIV RNA and F sequences were collected from participant BC021 at 8 time points between 2002 and 2005, prior to the start of suppressive antiretroviral therapy in April 2007 for this participant. And using sequences collected from each of these time points that have a known sampling date, together with proviral sequences, where in this case there were 93 proviral Neph sequences that we could study, we build a maximum likelihood phylogenetic tree relating the plasma and proviral Neph sequences to one another. Following this, we censor all of the reservoir sequences in the phylogeny and apply a root-to-tip regression approach, relating genetic divergence from the inferred root of the tree to sampling time for the pre-ART Neph sequences, such that based on their divergence from the root, we're able to estimate the date at which individual proviral sequences entered into the reservoir. And so when we do this for the plasma Neph sequences for BC021, we get an estimated root date, which represents the inferred transited founder event of February 12th, 2002, and an estimated evolutionary rate of 3.1 times 10 to the minus 5 substitutions per nucleotide site per day. When we use this regression model to infer the integration dates of BC021's proviral Neph sequences, we see that the majority of sequences entered into their reservoir in the period of time closer to ART initiation in 2007, where 68% of sequences entered into the reservoir in the 2 years prior to ART initiation. However, some older sequences were recovered, where the oldest sequence recovered was estimated to have entered the reservoir approximately 1 year after infection in early 2003. Meaning at the time of sampling, the sequence had time of sampling 2019, the sequence had persisted in the reservoir for 16 years. And this general skewing of reservoir composition towards proviruses archived there, closer to ART initiation, is more apparent when we scale these integration dates between estimated infection date and ART initiation, where here we can see that the vast majority of sequences entered into the reservoir in the latter half of this individual's untreated infection. And when we apply this approach to other participants in our study, we see that the skewing of reservoir composition towards those sequences archived there closer to ART initiation is relatively consistent across participants. Although in each individual, older sequences that have persisted for longer and were archived closer to time of infection were also recovered. An exception to this is participant BC004, where the reservoir was comprised of proviruses that were archived there throughout untreated infection. Although this individual differed from others in our cohort in that they were treated comparatively early in infection, within 2 years of their estimated infection date, while all other study participants were well into chronic infection at the time ART was initiated. So something that we're really interested to know is that since decay rates of proviruses with different genetic features have been shown to differ, and proviruses with different defects vary in their ability to express RNA and proteins, whether the age distribution of proviruses with specific genetic features also differs from one another. What we found when we combined data across individuals is that in fact the age distribution between intact proviruses and those defective due to 5' defects, large deletions, inversions, premature stop codons, scrambles of genes, or that are human HIV chimeras does not differ. And so what this suggests is that proviral integrity is not the most critical feature that modulates proviral longevity after many years on ART. So in summary, defective proviruses dominate the persistent reservoir, where individuals differ with respect to the burden of specific defects and clonality therein, and where clonal sequences are enriched in the intact and 5' defective reservoir subsets. In most individuals, the majority of proviruses that persist in the reservoir following long-term ART were archived there closer to ART initiation, although this is not universally true across participants, and within participants, sequences from early in infection can be recovered. And proviral age distribution does not differ by genomic integrity. And so this is important because it really highlights that the persistent reservoir in each individual is unique and diverse, where HIV cure approaches will need to take this into consideration. And also highlights that further investigation into the features promoting proviral longevity following long-term ART is required. And for acknowledgements, thanks very much to all of my co-authors who made enormous and invaluable contributions to this work, and also to our study participants without whom we could not do this. To our funders, and finally to the conference organizers for giving me the chance to speak today. Thanks very much.
Thanks very much, Natalie. That was stellar, as always, very well presented. Excellent talk. Questions from the audience or even from other panelists, perhaps for Natalie? You can stop sharing, Natalie, if that's easier.
Okay, I'll leave it just in case it,
okay,
for a moment, and if it's helpful for questions.
Yeah. Maybe I'll go first just to get the crowd warmed up here. Perhaps I didn't miss I may have missed this at the beginning, but in relationship with the talk from this morning, did you have elite controllers? You probably knew that was coming, right? Or elite controllers in
No, so there's none of the 6 people in our cohort are elite controllers, but yeah, the talk this morning definitely gave lots to think about in the context of data.
Understandably.
Yeah.
Any interesting elements, perhaps, that you and your team uncovered from the Neph sequence analysis that's shareable? I mean, anything interesting there, perhaps, or like the actual composition of the amino acids, I'd say.
Ooh, I haven't looked in detail,
okay.
So I guess I can't comment right now. Great. If the Neph sequences themselves are, there's anything special about them.
Let me just see if something is. I think Mitch Mumby has a question. Mitch.
Oh, sure.
No, Jimmy, you took my question. I was going to be about looking at genetic diversity, because we're kind of interested in some of the aspects of latency. We're looking more at the functional angle. So yeah, I was going to ask more about the genetic aspect of it, because we're trying to peruse through sequences ourselves and pull something out of it. So I was just wondering, but if you can't speculate too much on that, then I totally understand.
Sorry.
Okay. Thank you for pitching in. All right, let me just look at the message board here. Oh, there's a question from Alan Cochran, I believe. Interesting observation, but how do your observations fit with the data from monkeys that the reservoir is established very early after transmission? Any perspective there, Natalie?
Yeah, so I guess like we do, like both in monkeys, like Alan Cochran suggests, and in humans, we know that the reservoir begins to be seeded very early in infection. To some extent, our data does support that as well, because we are able to recover proviruses that have stuck around for in the order of 20 years that are from close to when the individual became infected. But what we also are gaining an appreciation for is that the reservoir is not static, and just as soon as a provirus enters into the reservoir, it doesn't necessarily mean it's going to stay there. And there's evidence now that proviral turnover during conditions of active infection happens and is actually faster than when individuals are on ART. So
okay. Well, let's all thank Natalie for an outstanding presentation, and we will move on to our next presenter. Okay, thank you. Our next talk is by, just a second here, Anne Monette from the Lady Davis Institute, McGill University, on HIV-1 biomolecular condensates. Anne? Oh, maybe unmute your mic, Anne, just before you're on. Perfect, thank you.
Hi everyone, thank you, Jimmy. Bonjour tout le monde. Let me first express my gratitude for the opportunity to present some of the work that we're doing in an exciting new phase of fundamental research. We now appreciate that most cellular processes are nucleated by biomolecular condensates and the membraneless organelles that they generate. What we know is that biomolecular condensates, or BMCs, are formed by liquid-liquid phase separation of disordered proteins that bind metals and nucleic acids. And while this rapidly growing field is looking for the holy grail of their therapeutic potential, we and others have discovered that numerous viruses also use biomolecular condensates for their replication. So before phase separation was a thing, many terms were historically used to describe viral BMCs, including viral factories and inclusions, virosomes and viroplasms, and mini-nuclei and agrosomes. We also used to describe these RNA protein condensates or co-condensates as ribonucleoprotein complexes. From their most recent investigation or reinvestigations and reclassification, and now as viral BMCs, we are learning that these can nucleate viral transcription, replication, assembly, and packaging. But we've only scratched the surface and don't know how we can target them clinically. The bottom row of this schematic shows our group's contributions demonstrating that HIV uses these BMCs for general virus assembly and release. So I'll first summarize our published findings, or recent ones, on points 1 and 3, that pan-virus RNA binding protein motifs are juxtaposed by disorder domains, and that pan-virus zinc-dependent nucleocapsid condensates mediate the late stages of replication. Then I'll show you the new data on points 4 and 5, demonstrating that HIV-1 nucleocapsid is the central scaffold of infecting HIV-1 BMCs, and then that HIV viruses become BMCs themselves from protease-mediated GAG maturation. So initially we had published that HIV-1 nucleocapsid, or NC, could induce stress granules. These were formed by wild-type NC, but not zinc finger mutants, and could be depleted by zinc chelation. NC-specific zinc ejectors cause aberrant viral RNA localization, while mutating NC zinc fingers ablates HIV's ability to block arsenite-induced stress granules. We discovered that this was due to the disorder of NC's ability to phase separate into zinc condensates. And this was also true for divergent pan-retrovirus NC and GAG proteins. Later, our meta-analysis demonstrated that this was likely the case for NC and GAG-like proteins across many unrelated viruses. And this was due to their disorder domains juxtaposed with metal ion-controlled RNA binding motifs that are also the hallmarks of pathological aggregates of neurological diseases. While we concluded that phase separation by the NC domain assists virus assembly, it was a mystery that while NC induced stress granules, GAG is well known to block them. This dichotomy could be explained maybe by the fact that GAG is found in cells and in immature viruses, while NC is found in mature viruses. So we hypothesized that this virus maturation by protease-mediated cleavage of highly ordered structural GAG and GAG-POL introduces these smaller bits of disordered proteins that generate HIV-1 biomolecular condensates. And so protein disorder scores were great predictors of the propensity to phase separate for capsid, reverse transcriptase, integrase, and protease. Yet NC recovers much faster in FRAP assays since its higher relative disorder should make it the most liquid-like. Then when inner core proteins were fluorescently labeled and transfected into cells, these formed dynamic NC-centric hexanediol-sensitive and spherical micron-sized condensates that exhibited fusion and fission. When testing numerous clinical antiretroviral drugs, the most striking result was that from the original specific NC zinc ejector, seen to rapidly immobilize and dissolve NC co-condensates. And so this positions NC as the central scaffolding condensate and suggests that HIV or infectant HIV viral cores could be pharmacologically targeted via zinc ejection. So labeled viral RNA also most readily condensed with nucleocapsid and to a lesser extent reverse transcriptase, as we could expect within virus cores. While HIV BMCs are visible in both cytoplasm and nuclei early after transfection, this is also true for DRCAT and primary CD4 T cells. We also frequently observe dynamic co-condensates linearly trafficking along visible structures like pearls on a necklace. And so Tubulin Tractor was able to establish that co-condensates are trafficking along these microtubules, as has been formally shown for infecting HIV cores. So to loop back to testing the hypothesis of how protease induces HIV-1 biomolecular condensates, when labeled GAG and viral RNA were transfected into cells alone, they didn't readily co-condense or undergo condensation. But if protease was added a few hours later, then they could undergo efficient condensation. And protease activity was validated by Western blotting. Protease mutant expression constructs showed us that GAG-POL could also not condense on its own without active protease, and again generated these hexane diol-sensitive condensates. These findings are actually quite novel, making us speculate that virus maturation creates these HIV-1 biomolecular condensates. And so now for sort of the pièce de résistance of today, to further prove that virus maturation creates biomolecular condensates, we did this, we in vitro mixed the core proteins and the viral RNA, representing the components of the pre-integration complexes that are trafficking from plasma membranes to nuclear sites of integration. And our analysis shows that these core proteins aren't randomly mixing at all. They're rather quite spatially organized. So in fact, to our surprise, these in vitro generated condensates or co-condensates or BMCs possess this striking resemblance in architecture to HIV virus cores. And to our knowledge, this is the first demonstration that HIV proteins self-assemble with defined geometry, resembling viral cores, and is quite strong experimental evidence that mature HIV viruses may themselves, in fact, be biomolecular condensates. So we hope that the next great review will add that the early stages of HIV replication also utilize phase separation, and that mature HIV virus cores are themselves NC-mediated biomolecular condensates. We're now working on that central step to prove that phase separation is also involved in HIV replication and translation. So I'll leave you with our perspectives that viral proteins are much more disordered, as we've seen today, than their eukaryotic counterparts. And the harder we look, the more we may find that viruses are constantly using phase separation, and that some viruses may themselves be bona fide biomolecular condensates. And while we've all been very busy with COVID, it's really, I love that we're still striving to find creative ways to cure the world of the HIV/AIDS pandemic. And with that, I thank the amazing Andrew, May for her technical perfection, Maya and Rob for providing us with a lot of these proteins that we labeled and put into cells, our collaborators throughout these projects, and for this wonderful audience. I'd be happy to answer any questions you may have.
Thank you very much, Anne. That was a stimulating, excellent talk. Very well presented, interesting data, thought-provoking. Okay, we have a question here, I believe. Let me first get a question from the room. This is from Art Poon. Fascinating study with an exclamation point. Which intrinsic disorder predictor did you use to generate disorder probabilities?
Oh, we typically use Ponder.
Ponder.
Yes, which
Okay.
is.
Okay, thank you.
But if you have another one to suggest.
Yeah, I'm sure. Because I guess his next comment is that he and his group, they found substantial variation in accuracy for the different predictors. So potentially some feedback there from Art Poon. Okay. I was also quite fascinated, and myself, with the the strings on the necklace image, right? And then of course you had the tubulin under. Have you had the chance yet to do the experiment with the tubulin inhibitor? What would it be? Nocodazole? I don't remember.
So we have also done experiments with tubulin or microtubule inhibitors. So we've applied both nocodazole and we've applied arsenite as well, okay. Which causes the different localization of these biomolecular condensates, arsenite to perinuclear regions, and right. Nocodazole dispersed away from the perinuclear regions.
All right.
Yes.
Just maybe one more while people think of more questions. In general, though, I mean, when we think of, the zinc binding, how generalized do you think it is or not to divalent cation binding? I mean, I'm just curious. Do you think other metals can bind too?
Yes, I think that actually there's quite a lot of competition between metals for these proteins. And the conclusions from the viruses study or the meta-analyses that we performed was that there was a stronger competition between zinc and copper than any other. And this is quite well established in studies across the literature for neurodegenerative diseases that are promoted by phase-separating tau and TDP43 and FOS and all of these other proteins that aggregate when unregulated.
Okay, very fascinating model at the end, an image that was really spectacular. Let's thank Anne for a great talk.
Thank you.
Thank you very much, Anne. Okay, so our next presenter is Subha Dahal from the University of Toronto. Okay, and Subha will tell us about antivirals for HIV and coronavirus replication. Subha, go ahead.
Can you see my screen?
Yes, we can see it very well.
Okay. Hi, everyone. Let me get my laser pointer. Sorry. Okay. Hi, everyone. I'm Subha Dahal from Cochrane Lab at UFT, and today I'll be talking about a small molecule kinase inhibitor, harmine, that I've shown to possess antiviral activity against two completely unrelated viruses, HIV-1 as well as coronavirus. Viruses, being obligate parasites, depend on multitude of host factors or pathways for their replication. And HIV-1 is a prime example of the virus that relies on host splicing for its protein production. And one of the key players of splicing are SR proteins, whose phosphorylation is critical to their function. SR kinases belonging to SRPKs, CLEX, and DARK1A families of kinases phosphorylate SR proteins, which regulate the function of these proteins in different steps of RNA metabolism. And given the dependence of HIV-1 on host splicing, we asked, could we exploit the modulators of this processing as novel anti-HIV agents? To answer this question, we screened inhibitors of host SR proteins and SR kinases for their activity against HIV-1 in HeLa cells containing doxycycline-inducible HIV-1 provirus in them. And compounds in our screening included TZ003, KHCB19, leucettin L41, and harmine. Screening in the HeLa cells identified one hit compound, harmine, that was able to inhibit HIV-1 structural protein envelope as well as GAG and the viral regulatory protein. And on the right is the quantitation of the Western blots across at least 6 independent assays. Dose response and cytotoxicity assay with harmine showed dose-dependent inhibition of viral GAG expression. And at the dose used in the cell system, which is 10 micromolar, there is minimal cytotoxicity with CC50 above 50 micromolar. The compound also altered all classes of viral RNAs, reduced TAR transcripts unspliced as well as singly spliced transcripts with significant accumulation of multiply spliced HIV transcripts. We also examined the effect of harmine on primary targets of HIV-1, CD4 positive T cells, as well as macrophages. These cells were obtained from HIV uninfected healthy donors and infected with wild-type HIV-1 89.6 and treated with DMSO or harmine or the combined antiretroviral therapy Ipivir Kaletra for 3 days, after which the cells were assessed by flow cytometry for percent viability and percent infection. And you can see that in either CD4 positive T cells or macrophages, neither Ipivir Kaletra nor harmine had any viability issue relative to DMSO control. And harmine treatment resulted in 50% reduction in percent infection in CD4 positive T cells, but in the context of infected macrophages, harmine had limited impact. But over the course of 10-day treatment, unlike the antiretroviral therapy Ipivir Kaletra, harmine treatment reduced GAG mean fluorescence intensity, indicating reduction in viral antigen in already infected cells. We then investigated into the known targets of harmine in the context of blocking HIV-1, the SR kinase DORC1A, as well as the enzyme monoamino oxidase A. We performed DORC1A knockdown without or with harmine treatment on one side, and in parallel utilized monoamino oxidase A inhibitors, harmine and moclobamide, for their effect on HIV-1 gene expression. However, we did not see any anti-HIV-1 activity with DORC1A knockdown without harmine treatment or with the monoamino oxidase A inhibitors. But anti-HIV activity was retained despite loss of DORC1A and harmine addition, indicating that harmine might be mediating its anti-HIV activity by targeting some other proteins or pathways. To determine if harmine could be impacting other SR kinases, we examined its effect on SR kinase expression levels by Western blot in CD4 positive T cells treated with DMSO or harmine. And interestingly, you can see that harmine treatment increased CLIC1 kinase levels by 8 folds, which has been quantified over here. And reduced CLIC2 kinase level by 25% with limited impact on CLIC3 or SRPK1 kinase levels. In addition, with the CLIC depletion assay, we have shown that CLIC1 and CLIC2 expression levels have radical effects on HIV-1. I don't have the time to show the data, but you have to trust me. We show that CLIC1 depletion enhances HIV-1 gene expression, while CLIC2 depletion is antiviral. And this data further suggests that harmine might be mediating its anti-HIV activity through relative changes in the abundance of CLIC1 and CLIC2 protein levels. In parallel, with the intense research to beat the current COVID-19 pandemic, we also screened all these compounds initially against human seasonal coronavirus strain 229E, which belongs to alpha coronavirus genera and causes common cold. As indicated in the schematic here, HU7 cells were infected with 229E virus for an hour, after which the virus was removed and cells were treated with compounds and soups harvested 24-hour post-infection to assess a viral genomic RNA release in media by qPCR, and toxicity assay was performed on cells by Alamar Blue. Again, our screening identified harmine as a potent hit to inhibit seasonal coronavirus strain 229E with more than 95% reduction in virus particle release in media. And subsequent dose response curve showed dose-dependent inhibition of 229E genomic RNA release in media with the increasing concentration of harmine, and also limited impact on host cell viability with a CC50 above 100 micromolar. And interestingly, harmine concentration at 25 micromolar resulted in complete inhibition of extracellular viral genomic RNA, which is a measure of viral particle release. And consistent with the loss of viral RNA release, we also observed loss of intracellular viral genomic as well as total RNA. As indicated in the representative Western blot, harmine treatment also resulted in around 80% reduction in 229E nucleocapsid protein, which is one of the structural proteins of the virus. After success in seasonal alpha coronavirus strain, we moved towards pathogenic SARS-CoV-2 SB2 strain, which belongs to beta coronavirus genera. And you can see that harmine treatment of SARS-2 infected HU7 cells resulted in dose-dependent inhibition of SARS-2 particle release in the media. And consistent with the loss of particle release in media, around 60% reduction in SARS-2 nucleocapsid protein levels was observed. Next, to decipher the stage of coronavirus replication impacted by harmine treatment, we performed time of addition assay. So in this assay, cells are first infected with the virus, and harmine is added at different time points. The time points are 0, 12, and 16 hours post-infection, and viral soups are harvested at 24-hour post-infection. These time points for this assay was determined by studying the replication kinetics of the virus, where we found out that the viral and protein, or the nucleocapsid protein, starts forming at 12-hour post-infection and peaks until 24-hour, and the particle release starts at 16-hour post-infection and increases up to 48-hour post-infection. So after selecting these time points, we performed the experiments, and you can see that delaying harmine addition up to 16-hour post-infection is as effective as adding the compound immediately post-infection, which indicates the compound to be a strong post-entry inhibitor of coronavirus replication, and further indicates that the compound is able to arrest virus replication even after the onset of the structural protein N. In summary, we have identified a kinase inhibitor harmine with activity against two completely unrelated viruses, HIV-1, which replicates in the nucleus, and coronaviruses, whose entire replication takes place in the cytoplasm. Harmine-induced changes in SR kinases indicate that the compound might be mediating anti-HIV activity through relative changes in the abundance of CLIC1 and CLIC2 kinase level. And we have also done RNA-seq experiments in CD4 positive T cells treated with harmine, and at the dose required to inhibit the virus, at least in case of HIV-1, the compound has minimal impact on host gene expression or host alternative splicing events. We observed only around 2.7% of the genes to be differentially expressed at absolute log fold change greater than or equal to 1, and around 1.2% of genes differentially spliced at delta PSI greater than or equal to 10%. And together, our data establishes the feasibility of manipulating host cellular processes to control the replication of multiple human pathogens of concern. Now, with that, I'd like to thank my supervisor, Dr. Alan Cochran, who has been immensely supportive, my lab members, our collaborator, CAHR, funding agencies, and very thankful to CAHR for selecting my abstract for oral presentation and giving me this platform. And I'm happy to take any questions.
Thank you very much, Suba, for that very interesting talk. Very well presented and great data. Well done. Any questions from our panelists or perhaps from members of our audience? I know you're out there and I know you have questions, so please just post them on the board or just pick up your hands and let us know. Okay. Just before that, perhaps, maybe I'll go for the first question, Suba. Pretty interesting, right? And I mean, obviously you have these effects with the two different viruses, which is quite fascinating. How would you reconcile that, you know, you're getting two different targets from the same compound, I guess, right? So
so we know that HIV-1 relies on host splicing. And though there is no direct dependence of coronaviruses on splicing, studies have recently shown that the coronaviruses nucleocapsid proteins have SRRS-like motifs, which are phosphorylated by the host kinase, SR kinases. And this phosphorylation is important for the early stage infection by the virus. The phosphorylation allows N protein unfolding, and unfolding of the N protein will allow the viral RNA to dissociate so that it can translate to viral polyproteins and initiate the early stage of infection.
Okay. And do you think that would these sites be equivalent or similar between seasonal coronaviruses and pathogenic SARS-CoV-2 from what you've seen?
Sorry.
Do you think there'd be similarities in those sites, at least for the coronaviruses, between the seasonal coronaviruses and
so
SARS-CoV-2?
So these SRRS motifs are conserved across all the coronaviruses.
They're conserved.
They have
okay.
been found. Yeah, they have been found across all the coronaviruses. But though the receptors are different between the seasonal and the pathogenic coronaviruses, we are seeing the same effect.
Okay.
So it could be.
Are there higher concentrations where you're starting to see potential toxicity from harmonium? I mean,
so there are animal studies. So there are animal models where they have used this compound, and there is no toxicity in mice up to 100 mg per kg. And the interesting thing about harmine is it's present in our diet. So we have been consuming this in nanogram to milligram quantities through our daily dietary intake. It's present in coffee, tea, rice, wheat, bread, meat.
That's great. I'll keep drinking coffee then and everything else. All right. Okay, here's a question. All right, let me just read it for you. While you didn't see major short-term viability issues in your cellular models, do you expect to see any issues with viability in animal models?
Yeah, as I mentioned,
just answered this, but go ahead.
Yeah, I think I just answered that question. So the LD50 in mice via IV route has been shown to be around 38 mg per kg body weight, and the LD50 in mice via the subcutaneous route is higher, like it's 200 mg per kg body weight. So there have already been experiments done. However, the compound is not yet clinically approved for use as a single agent in humans.
Okay. And there is a follow-up question here. Is there any interest in testing in macaques, perhaps?
Sorry,
macaques, non-human primates, to test it in non-human primates.
Non-human primates is like, macaques.
Yeah.
I don't think I found the data. I mean, maybe I should like.
Yeah, I guess maybe they're suggested in the future, right? So I mean, and I don't know who this is because they're listed as anonymous. I'm not sure if they're proposing it to you, but maybe they'll reach out.
Okay, but I don't think it would be that toxic because it's in our daily diet.
Okay, all right. If there are no further questions, let's thank Suba for a fantastic talk. Thank you very much.
Thank you. I'll stop my share.
Okay. We are now at the 8th and final speaker, but of course not the least. I'd like to present Amit Gaba from the University of Saskatchewan. And Amit will tell us about HIV transmitted founder VIF proteins and how they potentially induce or not degradation of ApoBec. Amit, whenever you're ready.
So I believe you can see my screen.
Looks good on our side, Amit. Go ahead.
So good afternoon, everybody. The ApoBec3 family of cytidine deaminases in primates and humans has 7 members that act as host restriction factors against retroelement retroviruses and some DNA viruses. The family includes 5 members that have been reported to inhibit HIV in absence of its VIF protein. In the absence of HIV-VIF, ApoBec proteins may get incorporated into the budding virions along with the viral RNA. When these virions infect new target cells, the ApoBec3 proteins may prevent successful viral replication by both DNase-dependent and deaminase-independent mechanisms. However, lentiviruses such as HIV have evolved a protein VIF, which interacts with A3 proteins, recruit an ubiquitin ligase complex and induces ApoBec3 ubiquitination and degradation. Ubiquitination of the ApoBecs usually occurs by K48 linkage, resulting in their proteasomal degradation. However, the ubiquitination can also occur by K63 linkage. This could affect the degradation of the A3 ApoBecs, as different type of ubiquitin linkage can lead to distinct events. For example, in one of our previous studies, we found out that for A3H, that is least sensitive to VIF-mediated degradation, VIF causes non-degradative K63 linked modification. VIF has several domains through which it binds ApoBac3 proteins, with some regions being specific for certain ApoBacs. Similarly, the SLQ motif of VIF promotes the interaction of VIF with allongin C and allongin B heterodimer. These interfaces have only been characterized by mutagenesis. As a result, we still lack the information on the full interface, knowing only the key points of contact. Also, previous studies the ApoBac proteins individually. However, the ApoBac3 proteins are co-expressed in CD4 positive T cells and have been shown to coordinately restrict HIV replication. So one of the study from our lab reported that ApoBac3G and ApoBac3F have the ability to interact, forming a heterodimer in the absence of RNA. This heterooligomer is more resistant to VIF-mediated degradation. However, the exact mechanism of resistance of the heterooligomer to VIF is still not known. Previously, HIV-1 VIF-induced degradation patterns of ApoBacs were identified using long-term lab-passage strains of HIV-1, such as LAI or NL43. But now we have access to the minimally passaged virus construct that were isolated from patient samples with 6 weeks of infections. These are known as transmitted founder viruses. The chronic control viruses are derived from patients who have been infected with HIV-1 for 6 months or more. So during one of our previous projects, we noticed that some of the transmitted founder viruses have differences in their ability to degrade ApoBacs. So we analyzed the VIF regions of these clinical isolates, and we noted several differences. However, the differences are not in the regions that are previously identified as important for interaction with the ApoBacs. This raises the question whether there are some critical amino acids that are still unidentified. Based on this background information, the question we want to answer are, is there a variability in the HIV-1 VIF-mediated degradation of ApoBacs by the clinical HIV isolates? Are there some key amino acids that remain unidentified? How A3s cooperate through heterooligomerization and how this affects restriction of chronic control and transmitted founder viruses? And what is the mechanistic basis of partial resistance to VIF-mediated degradation? To answer our first question, we did a VIF-mediated degradation assay as outlined in this schematic. As you could see in this figure, there is variability in the VIF activity of clinical isolates against A3G. ApoBac from some isolates, for example, isolate 42, 77, 850, etc., seem to be highly active against ApoBac3G, while VIF of others, such as isolate 185 and throw, is not so active against ApoBac3G. We saw similar variability in the activity of VIF of clinical isolates against ApoBac3F, ApoBac3H, and ApoBac3D. Here is summary of our results in the form of a heat map. The blue color here indicates less of ApoBac3 remaining after degradation and hence a more active VIF. A yellow color, on the other hand, indicates more of ApoBac3 remaining, indicating a less active VIF. Then we did the infectivity assay using TCMPL cells to see if the differences in the degradation activity of VIF translate into differences in the infectivity. In most of the instances, a more active VIF resulted in a higher relative infectivity and vice versa as expected, as we see here for isolate 42, which is more active, and isolate 185, which is less active. However, there were some exceptions also. For example, isolate throw, which seems to have less active VIFs, but still had higher relative infectivity in some instances. We are currently working on identifying VIF amino acids that are responsible for the observed variability in the activity of VIFs of these clinical isolates. Next, we focused our attention on our second question, heteroligomerization of apobacs and how this affects restriction of chronic control and transmitted founder viruses. As I mentioned before, since previous study was done with long-term lab-assessed strains, we want to see whether we see similar results with the clinical isolates and if there are any differences between the transmitted founder viruses and their chronic control counterparts. So for our experiments, we have used LAI SLQ delta VIF as control. The SLQ delta VIF produces a VIF, but the SLQ motif is mutated to alanine, triple alanines, preventing VIF interaction with alungin C, effectively limiting successful interaction with ApoBac3 enzymes. As you can see, the amount of remaining ApoBac3G in these ApoBac3G2 lanes is similar. However, when we compare ApoBac3F lanes, we can see a band when ApoBac3F and ApoBac3G are expressed together, but not in the lane where ApoBac3 is expressed alone, suggesting ApoBac3F is protected when expressed together with ApoBac3G. Similarly, in the virus plot, ApoBac3G is packaged in equal amounts. However, ApoBac3F is packaged more when ApoBac3F and ApoBac3G are expressed together. We then used the chronic control counterpart of isolate 470. VIF of the chronic control 470 has few differences in amino acids with the VIF of transmitted founder 470. However, as you can see, the results are similar for both transmitted founder virus and the chronic control for counterpart. Next, we quantified our blots and normalized the band intensities with tubulin. And as you can see, in case of ApoBac3F, there is slight protection for ApoBac3F from both transmitted founder and chronic control VIF when ApoBac3F and ApoBac3G are expressed together. To further confirm that this is not a strain-specific phenomena, we repeated the same experiments with other clinical isolates and observed similar results. That there is slight protection for ApoBac3F from VIF-mediated degradation when ApoBac3F and ApoBac3G are expressed together. The next question we want to answer is that the mechanistic basis for sensitivity and resistance for VIF-mediated, what is the mechanistic basis for sensitivity and resistance for VIF-mediated degradation? To answer this question, we focused our attention to ubiquitination. Our hypothesis is that the VIF variants that cannot disrupt ApoBac3 heteroligomerization lead to inefficient ubiquitination. To test our hypothesis, we are doing a co-immunoprecipitation assay as outlined in this schematic. As you can see, when I use the anti-V5 antibody for pulling down ApoBac3F, I could see a total ubiquitination when ApoBac3F is expressed alone or with ApoBac3G, but we could detect the K63 mediated ubiquitination only when ApoBac3F is expressed along with ApoBac3G. Unfortunately, our K48 antibody didn't work well, so I'm still in process of optimizing this assay. To conclude, there is a variability in the HIV-1 VIF-mediated degradation of ApoBacs by clinical HIV isolates. When ApoBac3F and ApoBac3G are expressed together, ApoBac3F is protected from degradation. The mechanistic basis of partial resistance of heteroligomer appears to be linked to the non-degradative type of ubiquitination. And a better understanding of the ApoBac3 VIF interface and exploration of the mechanisms resulting in the variability of ApoBac3 degradation induced by transmitted founder viruses will help in determining the contribution and consequences of ApoBac3 activity during a clinical HIV-1 infection. And the last, I would like to thank my supervisor, Dr. Linda Chalico, for her guidance and support. I would also like to thank my lab members for their help and support, and my funding agencies. I would also like to thank the organizers for giving me this opportunity to present my work. And I thank you all for your attention. I'm ready to take any questions.
Thank you very much, Amit. That was an outstanding presentation. Let me see if there's questions from the audience. Oh, I see one of the panelists has their, Mitch Mumby, go ahead.
Hi, great talk. I was just curious with your transmitter founder viruses, did you also look at the relative VIF expression levels between them? And if so, what differences would you expect to see?
Yeah, we have looked at their VIF expression, and it looks like they are not same all across the panel, but our next experiment will be to just use only VIF and the apobex together in a transfection assay so that we can overcome that variability and validate these results.
Just to follow up, and okay, that's really good. Do you plan to fuse fluorophores, or how do you plan to actually detect your VIF proteins for expression in your system?
Oh, we'll tag it. We have the tag versions
Okay.
of it. Yeah.
Okay. Any further questions from the audience or from our panelists? Anything you can say, Amit, perhaps on the sequence differences that you may have observed? You went a little bit quickly on that section there when you did these sequence alignments. Anything that pops up that you noticed or that you're pursuing or that you're interested in?
Yeah, so most of the regions that have already been identified are conserved in all these transmitted founders viruses, but the changes we observed are mostly at the boundaries of those domains that have been identified previously. So that is something to look at.
Do you think those regions may be more structurally linked, perhaps, instead of binding partners? Do you think they would affect the structure?
I think so.
Okay.
I think so. Yeah, we'll have to look at it. So.
Okay. All right. Thank you. Thank you. Any further questions? Let me just refresh this screen to make sure I'm not missing anything from the chat room. Okay. If not, let's thank Amit for an outstanding presentation. Thank you very much, Amit.
Thank you.
And let me take this opportunity to thank all 8 of the presenters today. These were outstanding talks. They were extremely well presented. I mean, fascinating stuff, very different subjects for some parts, very well presented. And of course, thank you all the attendees for attending, and perhaps Dr. Bernard, last word.
That would be great presentations. Congratulations for staying on time. We appreciate your work and just keep doing the good work.
Yeah. And Dr. Bernard and I would like to remind you about our special session tomorrow at 4:45 on
4:30.
4:30 p.m. Eastern time, and we do encourage you to attend on correlates of protection in people living with HIV that may be infected with SARS-CoV-2 as well. So I think you'd find that really interesting tomorrow at 4:30 p.m. Eastern time. Okay, with that, goodbye, good evening, good afternoon,
Yes.
wherever you are. Thank you.